Last section out of chapter 6 and 7 are the other applications of biotechnology. So these don't really fit specifically in human health or agriculture, but are different types of biotechnology that we are working on or have been using. So the first one I'm going to talk about is cloning. So this is creating organs or individuals from other individuals. So it's kind of the copying, photocopying version of organisms. So I'm going to take you through how we do this and then why we might want to do this. My question is, what was the first animal that we cloned? And the first animal that we cloned was a sheep. So that's why we're going to use a sheep for the example here. Cloning was first done successfully in 1997 by a scientist who cloned a sheep. So what we do when we want to clone an entire organism is we're going to take two of them. So like I said, we're going to use sheep. So we're going to take an egg cell from one sheep, and we're going to take the donor cell from the other sheep. In this case, it's a mammary cell, but we could take any cell. We're going to take the nucleus out of the egg cell. Why are we going to want to take the nucleus out of the egg cell? And I can give you a hint. We're going to use the nucleus from this mammary cell. So we're going to put it in there. Why would we want to take the nucleus out of the egg cell? Yeah, there's going to be too much DNA, right? This mammary cell already has its total 46 chromosomes, or actually for sheep it's another number, but you know what I'm talking about. It has the total number of chromosomes that it already needs. And if we left this one in there, it would now have 3 copies of everything, and that wouldn't work. We'll see when we talk in future chapters, just having a single copy of a chromosome causes a lot of problems. So having a full extra set would cause too many problems for this to work. So we're going to take these 2 cells then and fuse them together. We're going to fuse this mammary cell's nucleus especially together, but we're going to fuse the 2 cells together. So we're going to inject one into the other, so the mammary cell into the egg cell. Then we're going to zap it. We're going to give it a little electrical zap. This is going to hopefully combine the 2 cells together. If this step doesn't work, and this is one of the most common places where this fails, then nothing else will happen, right? But if this helps to fuse the mammary cell to the egg cell, then it will start to divide. Eventually, it will grow into an embryo, and so this is all done in a test tube or a petri dish, so this is all done in the lab. And when it gets to the embryo stage, then we're going to get another sheep. It could be the original sheep that was a donor. Often it's another sheep. And we're going to implant that embryo into the surrogate sheep. And if it works, then sometime later, we will get a baby clone sheep. And so that's how cloning an entire organism works. We do the same thing for any organism we wanted to do this with, right? You get a donor egg, donor cell, take the nucleus out of the egg, fuse these guys together, get them to divide, and then if it's an organism that needs a surrogate, then give it the surrogate, and then sometime later, you get the cloned organism. The thing is that this isn't always successful. In order to get Dolly, it took 272 tries. Yeah, 272 tries to get one cloned sheep. We're better at it today than we were in 1997 when they did Dolly. But, it's still not success every time. So it's not, every time we do this process, we don't get a clone every time. What other animals do you either know or think that we might have cloned? Because we obviously have done sheep, but what else do you think we have cloned at this point? So let's take a look. We have cloned quite a few different other species. So we've cloned mice, we've cloned cows, our sheep, we've cloned pigs, we've cloned goats, we've cloned cats, we've cloned dogs, and we've also cloned a primate. So one of our closest relatives as far as DNA goes. And the thing is, we are doing this for various reasons. So what kinds of reasons do you think we might be, for example, cloning cows? Why would we want to clone cows? And there's a reason why I picked cows. So, yeah, don't know. You might have said something like, to produce better beef, or if it's a dairy cow, if you have one cow that's really good at producing milk, if you make clones of her, you should get 5 or 6 cows that have really good milk production. I hope that makes sense. So that's one of the reasons why we're cloning animals like our farm animals. Cloning of pets, cats and dogs, that was tried first to see if we could clone those species, and now there are a couple of companies out there who will clone your cat or dog who has passed away. It's not inexpensive because this is an expensive process, and you do have to sign paperwork, stating that you understand that the cat or dog that you are having cloned, the clone will not be the same as the cat or dog that passed away. Because it will be genetically identical, so it will look identical to the, original pet. However, the personality will not be the same, because most of the personality, especially with our pets, is formed through their experiences, just like ours. All right, so that's full animal cloning. So then the question is, are there any medical justifications for cloning? So, for example, would we want to clone humans? All right. And I can tell you, we are not on the way to cloning full humans. If you've ever seen the movie The Island, where they have a whole island of clones that they harvest, we're not looking at something like that. Instead, what we're looking at is something like this. Producing what we call transgenic animals who contain human genes to produce human proteins like bacteria can. So we've already got our little hamsters that do our EPO, and we've got sheep that can produce some other medications that are used for treating cystic fibrosis. So we've already done this. So continuing to do this is incredibly helpful, because this can allow us to produce large amounts of human proteins that are not easy to synthesize, as far as, synthetically, artificially. So getting these guys to produce them, if we produce them in animals, they generally can produce them in larger amounts, depending on what type of protein we're talking about. Another one that we're looking at and working on are creating pig clones. So why would we want to do that? The idea is to use them for organ transplants. So I mentioned in a previous lecture that we have figured out how to use CRISPR to remove information from pig tissue to make it safe for transplantation. And so that's the idea for doing this. If we have genetically identical pig clones, we can CRISPR them as well to make sure that they are clear of all of that genetic information, and then we can just clone them to use them for transplant material. And then you've also got the possibility, and this is kind of where we want to go, not clone entire humans, but clone individual human organs for a genetic match. So like I talked about back when I was talking about membranes and, making sure that those fingerprints of the different proteins and so forth were matching on the cell membranes. Well, the best way to do that is to have your own cells making up that organ. So think about if we could take a sample of your own cells and then grow you a new liver or a new heart. So basically it would be cloning, but using your own genetic information. So that would be the cloning part. But instead of growing an entire human, just grow the organ that you need. And so that's where we're going with as far as the cloning part goes with humans. Is it possible to clone a dinosaur? If you're a fan of Jurassic Park, that's what they were kind of doing in the Jurassic Park movies. So is it possible to clone a dinosaur is my first question. And then my next question is, if it's possible, how do you think we could do it? What would we need to clone a dinosaur? So is it possible? No. There's a couple of problems here. The first problem is getting the DNA. So in the movies, they got the DNA from a mosquito in amber. The issue there is the DNA would probably be too degraded for the age. DNA breaks down very easily. And that mosquito probably didn't just bite a dinosaur, or at least didn't bite just one dinosaur species. So the first problem is getting the DNA. The second problem is finding a suitable surrogate. So the most closely related organisms on the planet to the dinosaurs are the non-avian dinosaurs. Yeah, that's right. The dinosaurs are still here. They are just the avian dinosaurs. These guys are the non-avian dinosaurs. I got very excited. The avian dinosaurs are things like birds. So the closest relative on the planet would be a bird. And the only bird that might be big enough for an egg for something like a T-Rex would be an ostrich. And we have no idea if the genetics would be anywhere close enough to make that work. If you watched Jurassic Park, they used something like frog eggs. So that's way, way different. You need to get the surrogate as close as possible to the original. In fact, all of our successful clones have been the same exact species. We haven't done any cross-species cloning like that before. I know. If I bummed you out on that, I'm sorry. All right, the last thing to talk about as far as other biotechnology is using DNA for identification. So you've probably, hopefully, I hope you've heard of DNA fingerprinting. So everyone has their own identifier as far as DNA. There's only one way to have genetically identical people on the planet, and that is if you are genetically identical twins. It's the only way you could get a DNA fingerprint that's identical. And you can see here the first individual, the first criminal that was brought to justice using DNA fingerprinting. So in 1987, Colin Pitchfork was convicted using his DNA. He was convicted of rape and murder using DNA evidence. What they did is what you can see over here on the right-hand side. DNA comes in all of those different sequences that we talked about previously. You've got, those introns, those sections that have just strange pieces and parts in them, as well as your genes. And what we can do is use this as an identifier. If you take a look at any individual's DNA, there are what we call short tandem repeats. These are in those intron sections, and these are exactly what they're talking about. They're short repeats of DNA sequences. So they are just repeated sequences over and over and over of the same sets of A, T, C, and Gs, whatever sequence that is. And they're found in the same region, but the length of the repeat changes from person to person. And so you can see here, mother and father chromosomes. So the chromosome from mom has a number of 3 repeats, while the chromosome that was inherited from the dad for this individual has 14. So this individual has what we call a 314 STR, because one of them is 3, one of them's 14. The other individual has a different one because their mother's chromosome has 5 STR repeat, and the chromosome from their dad has an 11. So they'll have a 511. So how can we tell what these are? Well, if we want to create a DNA fingerprint, what we do is look at 13 different STR regions. Each of these STR regions have a set of enzymes that we can use to cut out and then copy, so we can amplify them and make tons of copies so we can see them. Then what we do is put them in what we call electrophoresis gel. It basically is like thick jello. And you put an electric charge on it, so you can see it's got an electric charge here. DNA is negatively charged. So when you put the DNA sample on the negative end of the jello, the DNA is going to be pulled towards the positive end because negative and positive are attracted to each other, right? And it's going to get pushed away from the negative side. Now, the STRs are going to get sorted by their size because the bigger they are, the slower they move through the gel. The smaller they are, the faster they move through the gel to the other end. And so what happens is, as they get pulled into bands across the gel. And then we can read them on a computer, or we can read them as you can see here. So that's what these bands are. These bands are those STRs that have been measured out, right? They've been pulled across the gel and then measured. And so we can get something like this if this is a crime scene. So you can see we would take a specimen from the victim, so that way we have something to compare with as far as you can eliminate anything that would match them. Here's the specimen for the suspect that we know was there. And then we have the DNA fingerprints for the 3 suspects that they were interested in. And what you can see is that one of those individuals, the bars match up almost completely. There will be a little bit of wiggle, but they will almost be identical on a match. This will tell us that this suspect is very, very, very likely, and they can figure out the percentage, and that's why it says it was a 97% match or something like that, that suspect 1 was the perpetrator in this case, if it's a criminal type trial. We can also use these for things like paternity. So instead of victim and specimen, it could be mother and father. It would be mother and child and then father, the possible fathers. So this one would be the mother, this one would be the child, and then you'd run the possible fathers, and you'd look for the one that would be, in between, basically, the one that finished the profile for the baby, to figure out who the father was. So let's take a look. I do have a multiple choice question on this one. So using this DNA fingerprint, which suspect was present at the crime scene? And so in this case, it is suspect number 2. They're the ones that had the most matches. You can see suspect 1 does have a match here, and that's the only one that matches up. Suspect 3 matches on that one, matches on that one, matches on that one, but the other ones don't match. Suspect 2 is the one that has the highest number of matches. So DNA fingerprinting is another one that we've been using, like I said, since 1997. And it's much more reliable for evidence than, say, witness testimony. Witness testimony has been found to be highly unreliable, mostly because when we witness some sort of crime, we are usually under stress and we don't remember details very well. And so this is something that we can test over and over again and make sure that our results are valid. That's the end of chapter 6 and 7.